So previously, in the last chapter, we talked a bit about the primitive streak formation and the laying down of the notochord, and that's the very early precursors to the development of the spinal cord. And so now we're going to move on to look at the development of the neural tube, which is the next step in spinal cord development. So the neural plate and the neural tube, they come from the ectoderm layer of tissue. And there are three real main tissue types that arise from the ectoderm. So we have the surface ectoderm, which is primarily the epidermis. So this includes your skin, your hair, your nails, subcutaneous glands, sweat glands, etc. Your eyes, tooth enamel, etc. So anything on the outside, really. And then we have the neural crest, and at these cells lead to a lot of the peripheral nervous system, so the nerves outside of your spinal cord and brain, your melanocytes, so the cells that give pigmentation to you, facial cartilage, the more inside of your teeth. We'll talk a bit more about Schwann cells and glial cells in the future. And then we have the neural tube, which will be kind of the focus today. And the neural tube gives rise to kind of your brain and your spinal cord, right? So brain, spinal cord, your retina, which is kind of linked to your brain, motor neurons, etc. So the process in which this ectoderm layer becomes those three distinct ectoderm layers is called the process of neurulation. The embryo, once it undergoes neurulation, is called a neurula, which is spelled Neurula. So here we have kind of the views of neurulation in an amphibian embryo, which shows the early stage here, where you can see there's still the blastopore and still remnants of the primitive groove. And then we have mid-neurulation. You can see it's kind of starting to close, and we're developing this neural fold. And then the late neurula, where it's kind of the neural folds are fused, and we have a tube in the middle. So if we were to look at this from the transverse section as opposed to from a dorsal view, you can see we have the notochord, which is kind of underneath where the neural tube is going to develop, and this neural plate on top. And we have then these ridges forming, and we'll get into the kind of steps of this process as we go. We still have the notochord present, and then at the very end, this neural fold closes in on itself, and it forms an enclosed tube. So the neural plate lies on the surface of the embryo, as we saw in that dorsal view, but the nervous system will not lie on the outside of the organism. So there's got to be a way for this neural tube to make its way inside. So there's two types of neurulation. There is primary and secondary, and then there's this junction neurulation, which is kind of a third type, but it is just composed of a mixture of primary and secondary. So in primary neurulation, the cells surrounding the neural plate direct the neural plate cells to proliferate, invaginate into the body, and separate from the surface ectoderm to form the underlying hollow tube. So as you can see here, they proliferate, they invaginate down into it, and then it closes together, where the neural plate tube cells are then covered by the epidermis. In secondary neurulation, the neural tube arises from the aggregate of mesenchyme cells, into a solid cord that subsequently forms cavities that coalesce to create this hollow tube. So here we have these mesenchyme cells. They kind of congregate together into this tube, and then, or into a ball, and then the ball kind of hollows out and becomes a tube. So in the transition zone between the secondary and primary, it kind of varies among species, but it combines both mechanisms. So at which extent or how much of secondary and how much primary varies between species. But what this is you still see that invagination of some of those former epidermis cells that become neural tube cells. But you also have this combination of the mesenchyme cells making up the bottom of the tube. So you still end up with the same structure. So in this developing embryo, depending on the position of the tube within the embryo, if you're more caudal, which is more posterior, and or if you're more anterior towards the rostral gradient, you see the different types of neurulation. So at the very anterior side, you have primary. At the posterior side, you have secondary, and then in between the two, you have junctional neurulation. So here is kind of the process of neurulation of a chick embryo at about 24 hours. So what's important to notice from this slide is that neurulation happens kind of in a wave across the chick embryo, starting at the anterior end and proceeding to the posterior end. So as you can see here at the cephalic or the head region, we're starting to undergo neurulation, and you can see that the invagination and the closing of those neural folds is almost complete. But as we proceed down, we see that there is, this earlier and earlier stages, right? So here we haven't closed in yet. Down about halfway to the posterior, you see that we're forming these ridges, but they're not they're not circling around. And when you get all the way to the posterior, there is absolutely no evidence yet of this invagination or this closing of the neural fold. So this process starts at the anterior and works its way down to the posterior of the embryo. So neurulation has 4 distinct stages, but be careful because there's actually 5 steps. So stage 1 has an A and a B. So here we have stage 1A, and this is elongation. And so this is where the cells start to elongate. And remember, this happens in an anterior to posterior direction. And then still in stage A, but part B, we have folding. So we start to develop this MHP, which is the midline hinge point, and that's the point with which the folding is going to happen around. So stage 2 is the elevation of the neural folds. And so the neural folds are elevated as the primitive epidermis continues to move towards the dorsal midline. So these here, there we go, are the neural folds, and so they kind of rise up. And there's asymmetric constriction of actin in the apical side, changes the cell shapes to promote this MHP bending, so the mid-hinge point bending. So what happens, what I'm saying is that these cells change shape. The actin inside of them pulls to make them not columnar anymore, right? They're starting to kind of form these trapezoidal, I suppose, if I remember my geometry, trapezoidal shapes. And so that is what helps to bring this neural fold and this folding action together. So in stage 3, in convergence, we have the formation of another hinge point. This is the dorsolateral hinge point, and these cells start to form this wedge shape, and that brings the top 2 neural folds even closer together. So we have another hinge point that is able to kind of further squeeze around and allow this formation of the 2. And the last step here is closure, stage 4. And so at this point, the neural folds are brought together at the top here and adhere to one another. And if we look at the previous slides, there was this light blue region that was connecting the neural plate and the neural folds. and that's the neural crest. And these cells are just kind of the connector between those when we're still one solid piece of epidermis. But once the neural tube is formed, those neural crest cells will leave, they'll disperse. And so that leaves us with a neural tube that is not connected directly to these neural folds that will become epidermis tissue, or our epidermis tissue. We don't want our spinal cord attached directly to our skin. There's a little bit of a buffer in between, and so the leaving of these neural crest cells allows that buffer to exist. So different organisms have different closure points for the neural tube closure. And so in chick, we talked about the one closure point that proceeds from an anterior to posterior direction. But in mouse, we have three closure points. We have the cranio-ranchis closure point that starts kind of at the back of the neck and proceeds all the way posteriorly, but also proceeds anteriorly from that position. We have closure point 2, which is the anencephaly closure point, and that proceeds posteriorly and anteriorly from the top of the head. And then lastly, we have closure point 3, which is the anterior neuropore, which is kind of at the start of the mouth, and that proceeds only posteriorly. Now, if we were to look at human, it's very similar to the mouse, where we have the anterior neuropore at the mouth. That's closure point 3. We have the anencephaly closure point 2 at the top of the head, and we have the cranio-ranchis closure point at the back of the neck here. But in addition, humans have a fourth neural tube closing point, and this is at the hindbrain in the beginning of the lumbar region, and closure point 4. So it's just above that cranio-ranchis closure point, and it proceeds only in the anterior direction from there. So here we have a close-up of one of these closures, and as you can see, this is an example in these mammalian systems where the closure point happens at the middle, and both the bottom, the posterior, and the anterior neuropore are both open, and so this will zip up if it's one of the cranio-ranchis or the anencephaly neuropores that go in both, or closures that go in both directions. It'll proceed both posteriorly and anteriorly. So here we have a human embryo at the 10 somite stage. You can see the somites here on the side. And like I said, we'll get into those in the next couple chapters, what those develop into. But you can see they've labeled 3 of the closure points here. Here are the 2 behind the midbrain and the neck. You would have one kind of behind, the one at the jaw you can't see because we're looking at this from a dorsal view, and then you have that cranio-ranchis one in the center here. And here, as you can see, at 23 days of neurulation, the human embryo will have only this closed, or this open neuropore. Everything else is closed throughout the center here. So there's a closed or an open posterior and anterior neuropore, but for the most part, we've had closure all along this developing neurotube. So now we're going to talk a little bit about how this closure of the neural tube proceeds up in this zipper-like fashion. So here we see an early neurula, and you can see the stained blue here is the early formation of the neural tube, and you can see the folds kind of starting to come in from the outside and kind of close it off. And as you can see, as this proceeds up in an anterior direction here, that we start to close this tube. And at the very top here, you can see that we've proceeded from an open tube to this closed tube. And so we're going to get into a little bit of the details of how this works. So what happens here is, as this zipper is closing in this direction, we have these contraction points between the cells. And so the myosin is responsible for these contraction points, and it pulls the zipper point anteriorly to the next cell junction. So right here, in this little area here, that's going to contract. And we're going to pull this green portion to these points like this. And so as you can see in the next slide, now we have this green part, and we've pulled what was originally, let me change my color here, this junction to this black junction. And now what will happen is, as time goes on, what this green junction right here will contract itself. So the myosin in that junction will start to contract, and it will pull the next cells downwards. And so this will get pulled towards this junction here, and we'll have a green line here. And as you can see, if we look at this very first part of this step, we have, let me change my color one more time. We have 1, 2, 3, 4 cells that are attached to this junction point. And by the time we're done, we have 6 cells that have all been part of that junction. So that's how it continues to, it'll change this green attachment point up here. It'll contract, it'll become one of those red lines that is a myosin contraction, and it'll continue to incorporate and zip all the way up. So another aspect of this neural tube development is understanding how the neural tube separates itself from the surface ectoderm. And so when we talk about cell adhesion, like we talked about previously, this is based on those cadherin molecules. So we have 2 types in this case, N and E cadherin. And so the epidermis expresses E cadherin, and then the neural plate expresses N cadherin. And so when these tubes are pulled together in normal development, those N cadherin molecules will adhere to themselves because they have an affinity for other N cadherins, and the 2 E cadherin expressing tissues from the presumptive epidermis here, they will come into contact and adhere to them to one another. And so we have like attract to like, and then like attract to like in terms of the presumptive epidermis here. So we've talked a bit about primary interrelation, and that's when that folding occurs between those neural ridges coming together. And we briefly mentioned earlier secondary interrelation, and that's where those cells kind of migrate and form up to become a tube. So now we're going to talk a little bit more about the secondary interrelation. So remember, in a chick embryo, this secondary interrelation occurs in the caudal region of the chick embryo. And so here we have a 20-somite chick embryo, and in the beginning here, mesenchymal cells condense, as you can see right here, to form the medullary cord at the most caudal end of the chick tail bud, so very, very far posterior. So at a slightly more anterior position, here we can see that there are less of these mesenchymal cells because we're moving more up towards that junction position, and where we have this junction interrelation, which was the combination of both. In slide C here, we can see that the neural tube is cultivating and forming, and so in this one we have 2 different lumens present still, so they're starting to form, but we have 2 different interior cavities that have started to form, but that will eventually disappear over time. And then in slide D here, we have the finalized lumen. So as you can see, if we go from A to B to C to D, this kind of coalescing and formation of this tube from these disorganized cells. And what's indicated under my video here that you can't see is this is the notochord, is this dark ring below the forming neural tube. So let's talk a little bit about early brain development, which happens anteriorly to the spinal cord, obviously. And so here you can see we have the neural plate, and that is sitting on top of the notochord, which is probably not the best color here. The notochord, and then more posteriorly we have the primitive streak, and in slide B here you can see this in regards to the neural groove, which the label is under my picture here. So at the most anterior part of this chick, you see that the neural folds are beginning to converge here and move posteriorly past the somites and down towards the tailbone. And this is the beginning of the neural tube formation on the anterior side here. Remember, in chicks, there's only one closure point, and then it proceeds downwards. And then in pane D here, we can see that the beginning brain regions are starting to form. And so you can see that the kind of the neural tube runs within here, but these sections will develop into the hindbrain, the midbrain, and the forebrain. You can also see up here, this is the beginning development of the optic bulges, which will become retinas, and they will extend to the lateral regions of the head. So in the human brain development, we actually subdivide more. And so the forebrain becomes the telencephalon, the diencephalon, so that kind of gets divided into two, where the telencephalon will go on to become the olfactory lobe, the hippocampus and the cerebellum, cerebrum, which associates memory storage, smell, and general intelligence. The diencephalon, so the back part of the forebrain, is the optical vesicle, the thalamus, the hypothalamus, so temperature, sleep, breathing regulation, vision. The midbrain, which doesn't subdivide, the mesencephalon, but it's not two regions, is temperature regulation, emotional control, motivation. And then the hindbrain, which becomes the metencephalon, and the myelencephalon is kind of the ancient brain, as they say, which is muscular movements, reflexes, etc. So looking at the dorsal-ventral view, we have differentiation in the developing neural tube as well. And so early in neural tube development, the neural tube is made up of neuroepithelial progenitor cells that can be divided into these different color domains that we have here. And that is based on these unique complement of transcription factors in each domain. So in this blue region, dark blue region at the top, we have the expression of PAX3 and PAX7. And then in the most ventral region here, we have the expression of transcription factor NKX6.1. And then in the very central region, this green region, we have the expression of PAX6. And so these also form gradients. So these are the regions where they're the most highly expressed. And so the lighter blue and the yellow regions have combinations of the neighboring regions. So yellow will have both PAX6 and NKX6.1, and then light blue will have PAX3, PAX7, and PAX6. And so these gradients, again, are causing this differentiation along the ventral dorsal axis. So as this neural tube development continues, we get more and more genes that start to fire up, and more specificity on this dorsal-ventral axis. So as you can see here, we have a handful of genes depending on if you're on the most dorsal side or the most ventral side, LHX2, FOXD3, IS1 or ISI1, PAX2, etc. And so you get very specified cells here along this neural tube development. And so this also, they will start to create or interact with these periphery cells, cells on the periphery of the neural tube that will become neurons, motor neurons. So in the example here, MN will interact and be expressed in these external cells that become motor neurons, so they'll go and interact with muscle, etc. And so we need very different or differentiated cells from the ventral to the dorsal side, because all along this neural tube, different areas control different parts of the body, whether it be sensory neurons, motor neurons, etc. So here we have a histology view where each one of these different transcription factors are labeled with a different fluorescent dye, and as you can see, we have kind of this different sections of this developing neurotube that are expressing different transcription factors, and thus it will differentiate and have different cell types within this developing neural tube. So we know that different areas of the developing neural tube express different transcription factors, and that leads to these differentiations ventrally to dorsally. But how does this gradient arise? And as we talked about throughout the class, gradients kind of cause all of this differentiation. So when the neurotube is newly formed, we have the epidermis up on the top here, and we have the notochord below, and the epidermis expresses BMP4 and BMP7 and as a paracrine factor, so that kind of diffuses down to the neural tube, and the notochord excretes sonic hedgehog, SHH, and that paracrine factor diffuses dorsally. And so what happens then is we get the establishment of the roof plate of the developing neurotube and the floor plate. And once these are established, they will continue to secrete the paracrine factors that created them. So the floor plate will excrete more sonic hedgehog, which will diffuse dorsally, and the floor plate will excrete these TGF-beta family paracrine factors, which is BMP4 and BMP7, the ones that created the formation of this roof plate, but as in addition to BMP5, dorsal and inactivin, and that will diffuse ventrally. And so as these gradients diffuse, we've created this positional information, kind of like with caudal, and what happens then is if you have a high amount of sonic hedgehog, then you know that you are very much ventral. If you have a high amount of the TGF-beta paracrine factors, you know that you're the most dorsal. And if you have a mixture, you know you're kind of in the midline. And so based on that, those transcription factors that we saw in the previous slides are expressed. So here we can see a microscope slide where they have stained BMP4, which is the blue, and sonic hedgehog, which is the orange here. And as you can see, this is starting to form a gradient where BMP4 and the other TGF-beta paracrine factors are diffusing downwards to give dorsal positioning, and sonic hedgehog is diffusing dorsally to give ventral positioning. So here we see the relationship between the concentration of sonic hedgehog here at the bottom in nanomoles and the formation of these different sections of the neural tube. And so if you have a very high amount, 4 nanomoles plus, you develop into the floor plate. If you have between 3 and 4, you become the B3 section. If you're between 2 and 3, you become the motor neuron section, etc. And these are just stained based on their cell types over here to kind of give you perspective of the different regions of the developing neural tube. So again, this is an interesting experiment where if you notochord produces sonic hedgehog, which creates the floor plate, which then creates more sonic hedgehog, and you develop these motor neurons, etc., these different regions, V1, V2, etc., as we go more dorsally. And as proof of concept that the notochord is excreting the sonic hedgehog, which is causing the cells to differentiate into the floor plate and into motor neurons, if you were to take some notochord cells and put them kind of ventrolaterally to the developing notochord, it will influence these cells in the same way as it would when it's on the ventral side, and you get a second floor plate forming, and you get the same pattern where it almost thinks that in this direction is the ventral dorsal axis. And so it's just proof that the notochord is responsible for creating the floor plate and establishing this ventral side of the neural tube. So that does it for this chapter. I'm sure you're happy. It's a short one, 30 minutes. It's a record for least amount of time. And we will see you in the next chapter.